Hello from the German House in New York City, and welcome to Healthy Aging, Brain Controls of Longevity and Metabolism. I am Eva Bosbach, Executive Director of the North America Office of the University of Cologne. It is my great pleasure to welcome you all to our first Transatlantic Tandem Talk in 2022. This is the sixth talk in a series that we established on the occasion of the 10th anniversary of our North America office here in New York, showcasing various research topics from the University of Cologne in Germany and our partners here in the US. Today we will talk about healthy aging with Jens Brüning from the Cluster of Excellence on Aging Research CECAT at the University of Cologne and Andrew Dillon from our partner university, the University of California, Berkeley. Today's topic also continues a conversation that we started back in 2018 with our partner and co-host today, the German Center for Research and Innovation, also known as DWIH, here in New York, a joint series called Aging and Society. Questions around why we age and what exactly happens to our bodies and minds when we age continue to be of interest to all of us. We received over 600 registrations for today's event. Thank you for being with us. If you missed any of the previous Tandem Talks, you can watch them online on the website of the University of Cologne, New York office. Lastly, I want to thank all our wonderful partners for the Transatlantic Tandem Talk series, the German Center for Research and Innovation, DWIH New York, the German Consulate General, New York, the German Academic Exchange Service, the AAD, New York, Deutsches Haus at NYU, the Goethe Institute, New York, the German Embassy in Washington, D.C., America House NAW in Cologne, Wunderbar Together, 1014 Space for Ideas, and the German Research Foundation, DFK North America. And with this, it is my pleasure to hand over the floor to the program coordinator at the DFK office North America here at the German House, Dana Kolische. Dana?
Thank you, Eva. Hello, everyone, and thank you all for joining us today. I'm Dana Kelvishaw, and I'm speaking on behalf of the North American Office of the German Research Foundation, or Deutsche Forschungsgemeinschaft. The DFG corresponds to the National Science Foundation in the US. We offer funding to Germany-based researchers in any field of the sciences or the humanities for research projects of their own choosing. Historically, we've been very proud to contribute to the fact that the average quality of German science was quite high. On the contrary, the US historically focused more on just a few universities, but outstanding ones. In the beginning of the new millennium, German science policymakers looked for a way to combine these different approaches to have the best of both worlds, and finally landed on the excellence strategy of the German federal and state governments. One arm of this strategy is the funding of so-called clusters of excellence. The clusters of excellence stand for project-based funding in internationally competitive fields of research at universities or university consortia. Every year, the DFG spends a total of approximately 385 million euros for clusters of excellence. For the last round of applications in 2018, the Excellence Commission funded 57 clusters of excellence. It's quite an astounding number. Each of the clusters is funded by an average of 7 million euros per year for a period of 7 years. One of them is the Cluster of Excellence on Aging Research, CECAD, at the University of Cologne. I'm just as eager as you are to hear what Dr. Jens Brüning, PI of this cluster, and his colleague, Dr. Andrew Dillon, have to tell us about their groundbreaking work. With that, it's my pleasure to give the floor back to Eva Bosbach to introduce our wonderful speakers.
Thank you so much, Dana. I would now like to give a warm welcome to our speakers today. Please join me in welcoming Professor Jens Brüning of the University of Cologne's Cluster of Excellence on Aging, CECAT, and Professor Andrew Dillon of the University of California, Berkeley, who has also worked with CECAT as a member of the Global Faculty Program at the University of Cologne. Jens Brüning serves as director of the Max Planck Institute for Metabolism Research in Cologne and director at the Polyclinic of Endocrinology, Diabetes, and Preventive Medicine at the University Hospital in Cologne. His research focuses on the way our nervous system regulates our food intake and which brain cells are responsible for obesity and diabetes mellitus. Andrew Dillon is professor of genetics, genomics, and development, and an investigator at the Howard Hughes Medical Institute at the University of California, Berkeley. His work centers around the body's loss of control as we age and how this is communicated across brain cells. Together, they have published papers on the cross-section of their research, especially one in which they point out the extraordinary role the sense of smell plays in weight gain. They found that mice whose sense of smell was disabled did not put on weight, even when fed a high-fat diet. If their findings could be applied to humans, this could lead to a completely new approach in dealing with treatments of obesity. Imagine if we did not gain any weight just by preventing ourselves from smelling the food we eat. And maybe the pandemic sadly will give us some insights here as well. Jens and Andy, we look forward to learning more about your inspiring research, and thank you both very much for joining us today. I would now like to hand over to the moderator of today's talk, Katrin Di Paola, Program Manager at the DWIH New York.
Thank you very much, Eva. It's a pleasure being here. Hello from the German Center for Research and Innovation. I'm sitting one floor below the lovely ladies, Eva and Dana, and it's fantastic being part of such a powerhouse. And it's great, Eva, that we're continuing the Transatlantic Tandem talks on the topic of aging. As you mentioned before, it's a very hot topic still, and I think with 606 people registering who were hoping to find the secret to being forever young, I guess. But I'm also very intrigued by the idea that I can just stop smelling and stop, and then not gaining weight. So this is really, really cool. Before we start and before I hand over the floor to our presenters, first of all, thank you and welcome also from us, from the German Center for Research and Innovation. It's always great seeing you in your homes, in your offices, and taking us actually in your environment. And I have just a few points of housekeeping for you because we're a large group. So for now, we would kindly ask that everybody stays unmuted until the presenters are done with their presentations. And afterwards, of course, there's plenty of time still for a Q&A session. And there's two ways of asking your questions. Either you can put a Q in the chat, or you can actually type the question in the chat, and I will try together with Annabelle to filter as many as we can and to get as much answered as possible in the upcoming hour. And with that, I'm handing over to Andy, and I think we're all very, very excited to hear what's coming up.
Excellent. Thanks for the great introductions from everybody, and for the invitation to take part in this. It's always, a pleasure to do anything with Jens Bruning. We've been collaborators for at least 15 years, close collaborators. So it's great to come together in a joint session and, share some of the things that are going on between our labs and show you some of the intersection that's actually happening. It's also, it's great to hear that, the smell paper that we had was pretty profound. We would love to be telling you about that, but that is the work of a former postdoctoral fellow, Celine Riera, and that is her work, and she's taken it on to her lab. So I encourage you to look up her website and see what she's doing with it. Phenomenal scientists that collaborated with both me and Jens on that. All right, so as mentioned, my name is Andrew Dillon. I'm at UC Berkeley, and I'm going to start this off a little bit more basic, and then we're going to go a little bit more mechanistic when we move into Jens's talk and talking about neuronal control of aging and metabolism. So if I could have the next slide. Sorry, everyone, I don't have control of my slides. So I'm going to interrupt with each time asking for the next slide. So bear with me. So I'm representing the aging field, and this is a rather newer field for the research community. For the last 50 years or so, there's been mechanistic studies on the aging process. And this has been relegated primarily into studying aging of individual cells. And this is when we're talking about, anything as basic as the aging of yeast cells to how aging of human cells in a tissue culture dish are aging. And from this, we've learned some very fundamental key rules of how these cells age. First, we learned about the Hayflick limit of some cells have replicative limits on how many times they can divide, and this is determined predominantly by their telomere length. But there's other pathways that have been discovered in these studies as well that I'll cover throughout the talk. And I'll focus primarily on this concept of proteostasis. And that may be a new word to you, but I'll try to define it a little bit better coming forward. So keeping in mind, these are studies that are removed out of the entire organism. So on the next slide, while we've learned a lot about what happens in these individual cells, the question really needs to move into what happens when we move into complex organisms, metazoa. And so do the same rules apply? Is aging actually determined by these different rules that are set out? And if these rules do apply, does every single cell determine its own rule? You know, if you think about the human body with its trillion different cells, does each cell on its own determine how long it's actually going to live and what its actual route of aging is going to be? Or is there going to be some form of communication? And I think all of you are looking at this and saying, oh, duh, of course there's going to be communication. You know, we can look at this across the human population. We can see that, you know, people are aging in a coordinated fashion, that the liver is aging at the same age as their heart. You know, it's not that they're dysregulated. And so we're sort of hedging our bets that there's going to be coordination. And today we're going to talk a lot about, Jens and I, some of the coordination that we've uncovered. But please recall that you are only getting a snapshot of the entire aging field. There are many, many other researchers across the world that have looked at this problem, and there's lots of other data besides just ours. So keep that filter on when you begin to look at this. Okay, could I have the next slide, please? So I think it's becoming generally agreed there are some hallmarks to aging, and these may not be all of them, but this is actually a good representation of all the different processes that the field is generally beginning to agree upon that go wrong with the aging process. And through this, you can probably find your favorite aspect of biology. That's the beauty about aging research, is that it's so diverse that it encompasses almost every aspect of biology. Today, I'm going to focus a lot on proteostasis, which is how well the proteome is maintained across cells. But if you look at the top upper left, this altered intracellular communication, that is the communication across cells, we're going to marry those two together, and I think they're actually linked together to form one part of this pie and not two separate parts of this pie. So let's talk a little bit about proteostasis on the next slide. So there's lots of machinery in our cells that are there to take care of your proteome, to determine how well it's folded, how well it's maintained and kept pristine. And this is very obvious when we're young. We don't have diseases of protein misfolding mainly in the younger population. It mainly affects the older population. And so even though these misfolded proteins are made from birth, they really don't start affecting individuals until much later in life. And so there's processes that are there to monitor this. And they monitor, you know, the proteome of the mitochondria, the proteome of your cytoplasm. And the one that we're going to talk about most today is the proteome of your ER, the endoplasmic reticulum. So there are 3 different pathways that regulate this. I'm not going to go into details about all of these, but the one that you're looking at on the far left, the IRE1 branch, is the one we're going to focus on the most. And this is a very elegant system. It monitors misfolded proteins in the ER. And that IRE1 molecule becomes activated when there's misfolding. And its main job is actually to splice a message that's in the cytoplasm called XBP1 into a splice form, XBP1S, that can now be translated and as a transcription factor to go into the nucleus and regulate genes to come back and fix that folding stress that happens in the ER. It's a pretty phenomenal system. I'm sure you could ask yourself, I didn't know that there were messages that got spliced in the cytoplasm, and you're absolutely right. XBP1 is one of the only ones that is spliced in the cytoplasm. We can talk more about the evolution of that, why that could occur, but it may be some of this extracellular communication that I'm going to tell you about. So we can trick the system into thinking it has stress in its ER by simply overexpressing that splice form of XBP. So remember that in all of our experiments, we're going to go through and trick the system by merely overexpressing XBP1. All right, so on the next slide, we ask the question, is that we know that this response declines with age. So when we look at many different organisms across age, their ability to turn on this unfolded protein response of the ER declines with age. So the simple question was, well, can we simply restore it in old life? And if we can, do we get restoration of ER stress, but also do we have to do it in every single cell? And you would think a priori that you would have to do it in every cell, that every cell is worried about its own ER, and so it's going to restore each one individually of each other in an autonomous fashion. So we did this experiment on the next slide. We moved into an organism that was very simple for doing this. And this is the nematode C. elegans. If you're not familiar with it, It's an excellent organism to study the aging of post-mitotic cells. So this animal is only made up of 959 cells, and they are completely post-mitotic. So if it loses one of those cells, it can't replace it. There isn't a stem cell population for the soma in this animal. So it has some drawbacks, but it also has some advantages because it's very good for studying the aging of non-dividing cells. And there are other attributes here of why this is such a great organism. It's short-lived. And also the UPR that I showed you is the mechanics of it is completely conserved in the worm. And we know that that goes down. So on the next slide, we ask simply, when we restore XBP1 and we go across the 10 different tissues of the worm, we find that there's only one tissue that's essential for this. And this is the nervous system of the worm that's made up of 302 neurons, debatable 302, but around 302. And what we see is that when we overexpress this XBP1 as shown in red, it actually makes the animals super long-lived and they're very resistant to chronic ER stress. And so we're restoring this back to a youthful state and actually pushing them a little bit over the edge to actually make that happen. So this was a super exciting finding that Rebecca Taylor found when she was a postdoc in the lab. And the more interesting part of it is that we simply thought we're just protecting the neurons. And that's why these animals are living so long. But she did an elegant experiment where on the right side, she found that they actually had to communicate this ER stress out of the neurons into the peripheral cell types. And on the next slide, I'll show you some of this data just very briefly, is that there are different ways that neurons can communicate. They can either release small clear vesicles, shown by the mediation of UNC13, and also they can release dense core vesicles, which contain your biogenic amines, your neuropeptides, and hormones, and that's required with UNC31. And what Rebecca discovered is that UNC13 is absolutely required to communicate this stress. If you look at these animals on the top, you see the green, that's turning on the ER stress in the neurons, and in the bottom, you see it's communicating it to the tails. That's our reporter for showing this. And if you mutate UNC13, you see that we robustly turn off that peripheral signaling event. We also turn off the ability of these animals to live long. So it's not just protecting the neurons as sufficient, it actually has to be coordinated and communicated. And this is a very robust finding for us, and it's super exciting to uncover this. And so on the next slide, you can imagine there were three big burning questions that were happening in the lab. Is that what actually happens in the target tissue in the periphery when this signal comes out? Which neurons actually do this? Remember, we did it pan-neuronally in all the different neurons. Are there certain neurons that do it and certain neurons that can't do it? And then finally, what is actually the signal that's coming out of these neurons? And so I'm happy to report on the next slide that we've addressed most of these questions. Ryo Higuchi Sanabria was a postdoc in the lab and now has his own lab at USC, has uncovered a lot of these while he was in our lab. And you can follow it on the right side, these current findings, is that there are actually two branches of this unfolded protein response. One is the chaperone branch, the refolded misfolded proteins, but the other is a lipid modification branch to restore the membrane structures of the ER. Sort of makes sense that you have two different pathways going on. So what he quickly discovered is that serotonergic and dopaminergic neurons were absolutely essential for turning on this response. No other neurons really seem to play a role in this, but the serotonergic neurons and dopaminergic neurons seem to do this. What's also really neat is that the serotonergic neurons only turn on the chaperone branch of this. They both talk to XBP1 in the periphery, but the signals coming from the serotonergic neurons will turn on the chaperone branch, where the signals coming from the dopaminergic neurons will turn on the lipid accumulation branch of the UPR-ER. And what he uncovered is that definitely serotonin and dopamine are the signals for doing that. And now he's going forward in his own lab to understand, is there actually some form of cross communication? So it's amazing that in the periphery, XBP1 can receive different signals. And depending upon what that signal is, it'll either restore the chaperone functions of the ER or restore the lipid levels of the ER. So with that update, I want to just spend a few moments of your time telling you what is actually new in our lab and segue it into Jens's talk. So on the next slide, we were very blessed by having another postdoc in the lab, Dr. Ashley Frakes, who came to us as a glial biologist and made us aware that we were studying this phenomenal effect out of the nervous system, studying these neurons, but the brain is more than just neurons. And if you go to the next slide, you will begin to realize that glia, there are many different subtypes of glia in the nervous system. astrocytes, microglia, and oligodendrocytes. And actually, as the brain complexity goes up, as you go up through evolution, the ratio of glia to neuron actually increases dramatically. So in us, it's a 5-to-1 ratio, but in the worm, we're going to see it's more of a 1-to-5 or a 1-to-6 ratio. But also, glia are heavily involved in many of the diseases we associate with aging, especially metabolic disease, which Jens and I are going to tell you a lot more about. So we simply ask, do glia have a similar function as the neurons in regulating this response? And on the next slide, I will take you through some of this data of looking at the worm. And the worm is actually a great system to do this in because it's very refined. There's only 56 glial cells, 300 or so neurons, and we know the identity of each one of these different glial cell types. And more importantly, it's not clear that there's a different glial cell types. They more seem to be astrocytic-like, but they appear to be sort of like a primordial blood-brain barrier. So if you look at this nerve ring that is in the worm, it's actually ensheathed by these different glial subpopulations that are found there. And actually, as the axon goes out to the periphery and out to the outside environment, if you follow that along the worm, the glia follow it along as well. And not only does the neuron touch the outside environment, but the glia does as well. So there's opportunities for the glia to actually sample what's happening in the outside environment and transmit it to what's happening to the periphery. So on the next slide, we did the simple experiments where we took our magic trick of overexpressing XBP1S and did it in different glial subtypes. And on the left side in A, we put it in almost all the glia, around 50 of them. And what we find is the animals are robustly long-lived. And on the bottom, you can follow that they turn on this ER-UPR in the periphery. In the middle, we did the missing glia that were missing in experiment A, and those were not able to do this. So not all glia are equal in this process. And more importantly, on the right side, these cephalic sheath glia that encase that nerve ring and cover up the blood-brain barrier, well, not really a blood-brain barrier, but a barrier for the nerve ring, those seem to be very, very robust. And I have to point out that is only 4 glial cells total that we're expressing this in. It robustly extends that lifespan and robustly turns on this ER-UPR response. So we want to figure out what's going on with those cephalic sheath glia and maybe come back to why some of the other glial cells can't do this. So if we could go to the next slide, you may say to yourself, well, you're just doing the same thing that the neurons do, is that you're sending out the exact same signal, you're activating those neurons, the serotonergic and dopaminergic neurons to do the exact same thing, and you're just tapping into that system. So what we know from the neuronal system is it increases lifespan and stress. It's UNC13 dependent, so small clear vesicles, and it does lipid depletion, and it requires serotonin and dopamine to do this. And so what Ashley discovered is, yes, it definitely increases lifespan and stress resistance when we do it in these 4 glial cells, but it's UNC13 independent. In fact, it requires dense core vesicles. So it's doing it by a completely different mechanism, is that the neurons are sending out neurotransmitters of serotonin and dopamine, whereas it appears that the glial cells are sending out neuropeptides of one form or another. They both do the lipid depletion phenotypes, and we don't know the requirement, and it's not requiring serotonin to do this response. So on the next slide, I'll summarize what we do know that's happening here. So on the left side is our neuronal paradigm, where we cause ER stress in all the different neurons, or the serotonergic and dopaminergic neurons. And it requires small clear vesicle release through UNC13. And those neurotransmitters somehow act on these receiving cells in the periphery to make XBP1 turn on this ER unfolded protein response and make the animal live much longer. To our surprise, these 4 glial cells on the right side can also send out a signal to cause a very similar response in the periphery, but it does it very differently. It requires a processing enzyme to convert a peptide, a propeptide into a prohormone into a hormone using AGL3, which is a convertase that goes and cleaves a polyhormone into a single hormone. And then it requires UNC31 to actually package this and put it into vesicles to secrete it out. So I don't know why there are 2 different responses coming out of the nervous system, one that's neuronal and one that's glial. One idea that we have is that the neuronal one, using its neurotransmitter release, is very fast acting and responds, can go up and down very quickly, whereas the glial cells releasing its hormones may be very slow acting and be able to act at a longer distance. That remains to be determined, but we are converting these all into animal models as well. So on the next slide, I want to transition to the last two slides to set up Jens for his talk, is that, everything I've shown you is some really intricate signaling events that are coming out of the nervous system of an animal that only has 959 cells, lives in the soil. How real is it actually to be converted into vertebrates? And so Gökhan Hatamadzisill's lab has been a pioneer in looking at ER stress under conditions of high-fat diet and other conditions that are actually going to cause metabolic stress. And what he's uncovered is that overnutrition actually induces the ER stress response, and he sees it specifically in the brain, and there's peripheral effects that happen to the liver to change the release of insulin and actually cause blood glucose levels to be altered. More importantly, if you knock out XBP1, and this study that they did, they thought they were knocking out only neurons, but their driver actually knocked it out in astrocytes as well, actually exacerbated diet-induced obesity. So if you don't have XBP1 in the nervous system, the animals get super fat and super responsive to a high-fat diet. So our idea was that, well, maybe what we're finding in the worm is that maybe astrocytic XBP1 is protective against chronic ER stress. And one of those we would test would be, for example, a high-fat diet. So on the next slide, we created mouse strains where we could ectopically overexpress XBP1 in astrocytes, predominantly in the brain, but we have to acknowledge that there are astrocytes in the periphery as well. But we try to circumvent this by different stereotactic injections with other methods. But I'm going to show you this first experiment using a GFAP Cree mouse, which expresses in the astrocytes, and we're going to overexpress XBP1. And we're going to set this up and then have the animals a little bit later go onto a high-fat diet and see if they're actually going to be protected. And all this work was done in collaboration with Jens, not only when he was in a sabbatical here, but also in his lab in Cologne. So going with the next slide. What you see is in the red line, the wild-type mice gain weight when we put them on a high-fat diet. The black line is our control-fed mice. And in the blue is our animals that are overexpressing XBP1 for a few weeks before this challenge, and they've only been injected once before this. So we are seeing protection against the high-fat diet. It's not that they're eating less, they eat just as much. And if I went through and showed you all the body mass indices of all the different organs, you would see that the weight they're losing is all in their fat mass. So it's not a complete recovery, but we are well on our way to actually seeing this happen, that they are being protected. More importantly, we see improved insulin sensitivity and glucose tolerance. And so we think there is something coming out of these glial cells that are protecting these animals from a high-fat diet. On the last slide, I think I summarize what I've told you, is that remarkably in this worm, C. elegans, we see tremendous conservation going into at least the mouse, that when we turn on XBP in these astrocytic-like glial cells, we think we're seeing the release of a neuropeptide, and this is mediated by UNC31 and AGO3, to protect the periphery from the challenges of ER stress, one of them being, for example, in the mouse, a high-fat diet. In the mouse, we see this happening with glial cells in the brain communicating to the periphery, the hepatocytes, and the liver. We don't yet know the signal, and we don't yet know if it's UNC31 or AGO3 dependent yet, but we are actively working on that. I'd like to end, last slide, I think. Yes, by many people in the lab, past and present, that have worked on this ER stress project. And the stars, you'll see there are many graduate students that have contributed to this project and many, many postdocs there as well. And also a big thanks, Jens Bruning, he has 3 stars there, longtime collaborator with this, and we've had many help with many different labs throughout the course of this work. And I will turn it over to Jens, I guess now, or the moderator. Great, thank you, everybody.
Yeah, it's actually Jens' turn. Jens, go ahead.
Thanks, Andy, for setting the stage. And always a great pleasure, having the opportunity to, speak together with you. I hope you can see the presentation. Well, thanks, Eva, thanks, Katrin and Dana, for the kind introductions. And I also wanted to take the liberty to just take a few minutes to introduce to you CCAT Cologne, the Excellence Cluster in Cologne, give you a bit of the background how the whole initiative started and how it has developed over the last years before really going into the science of our lab. And so as Dana has pointed out, the DFG has put out this funding scheme, I think in 2006, the first time around. And so in 2007, we applied with the fundamental question, I mean, how could the Cologne community contribute to potentially societal relevant questions? And so one of the clear challenges of today's society is a demographic change. And what I put up for you is, what is pretty much known from the public is the ever increasing life expectancy. And what's shown here is a proportion of, people aged older than 65 at the population in Germany right now, and the projected increase to more than 30% in 2050. And that, in principle, of course, is a very positive development. However, it comes at the expense of an ever-increasing proportion of aging-associated diseases in the elderly population, both affecting the individuals as well as putting a pretty substantial burden on society to take care of these diseases. And I think, as Andy has really beautifully laid the ground for this philosophy, I would say, is that the aging biology field has really revolutionized our understanding that aging is a modifiable event. So it's not a given. You can modify it by dietary interventions or genetic interventions. And what is really interesting, if you study animal models, you can find if you delay the aging process, either by dietary or genetic interventions, you can delay the whole onset of the plethora of aging-associated diseases. So you can really, kind of shift the philosophy of how to treat aging-associated diseases. Instead of going for the pathophysiology of each and individual disease entity, if we understand how aging is determined, and if we can prevent aging as a fundamental mechanism, we could potentially devise new preventive or therapeutic approaches to cover the whole spectrum of aging-associated diseases such as diabetes, cardiovascular disease, Alzheimer, cancer, osteoporosis, let's say in one hit, instead of tackling each of these diseases individually. And this really we have put forth as our mission in 2007 to define the molecular and cellular mechanisms underlying the aging process, to define potential new targets for prevention, early diagnosis, or treatment of the whole spectrum of aging-associated diseases. And we are now in the third funding period of this initiative. And initially, we really started out, very much as Andy has laid out, that the field was very focused on cell-autonomous mechanisms. So what, dictates the aging of a given cell in the organism. And there are candidate pathways, inflammation, mitochondrial function, DNA repair. So very fundamental signaling processes in each and every cell, which can dictate the functional decline over age. And so during the first funding period, we've really focused on those cell autonomous mechanisms. But then moving on, we've really more turned our interest both to the cell-cell communication and the inter-organ communication in these processes. And more recently, also in line with what Andy has given you beautiful examples for, it's really not that our body operates in isolation from the environment. So we're constantly perceiving sensory input from our environment, views, smell. We're also communicating with the environment through microbes which live on our skin or in our gut. And so what we're also interested in to extend the inter-organ communication questions by how our organism interacts with the environment to ultimately shape the aging clock and the onset of aging-associated diseases. And I mean, this is an inherently complex research program. And so we've really brought together many groups within Cologne and the neighboring cities, and those comprise different institutes. So the core is the CCAT Research Institute, which houses many groups within the university, both from the medical and natural science faculty, which have found their common home there. Then across the street, the newly founded or recently founded Max Planck Institute for the Biology of Aging, the institute I'm working in, the Max Planck Institute for Metabolism Research, the university hospital to really allow immediate access also to patients and patient cohorts, and the German National Center for Neurodegenerative Diseases, DZNE, in Bonn and Cologne. So it's really a unified effort through multiple institutions, and the whole group of PIs now comprises more than 30 groups and more than 30 associated members. And as Andy is really a prime example, we've also put out the international faculty program to also have participate internationally leading scientists in the aging field, who unfortunately did not want to move to Cologne on this occasion. So, but still giving them the opportunity to work together with us. And so really coming to the science that is pursued in my group. So I'm a trained endocrinologist, and so my clinical interest for the last 25 years has really been on typical aging-associated diseases, such as diabetes and obesity. And I've just brought you the map of the states, highlighting the prevalence of these two diseases. And if I click the start button, you see how it continues over in 2-year steps, the ever-increasing incidence of overweight, obesity on the right hand, and the strong association with the ever-increasing prevalence of diabetes on the left-hand side. And this stops here at 2008, but the trend keeps going until actually now, and is predicted to ever increase further. So there is now more than 50% of the population being overweight, and the prevalence rates for diabetes are approaching approximately 10%. And really the fundamental question is what is really changing that we're gaining weight, and how does our weight gain lead to the onset of diabetes with all its devastating complications in so many patients across our societies. And so the classical view is, how is glucose metabolism regulated? I mean, that's a fundamental question in diabetes. So in diabetes, glucose homeostasis, the normal control of glucose levels fails, glucose levels go up. And the only hormone in the body which is able to lower blood glucose concentrations is insulin, which is released from the pancreas, which is outlined here. And it acts predominantly on the liver cell to suppress the new production of glucose, and on the fat cell and muscle cell to promote glucose uptake from circulation. So insulin does everything to lower blood glucose concentrations. And this is kind of the standard biochemistry textbook knowledge, that insulin acts on those three main tissues, liver, fat, and muscle, to lower blood glucose concentrations. However, what is interesting is if you really look into the body and look where could insulin act, you will find the insulin receptor, which is the docking protein for insulin on the cell surface, virtually expressed in each and every cell of the organism. So, and in diabetes associated with obesity, insulin does no longer act appropriately. So we term this insulin resistance. Despite the pancreas releases insulin, insulin fails to appropriately lower blood glucose concentrations. And all the research efforts really so far had been focusing on those main classical insulin target tissues, liver, fat, and muscle, to understand why glucose concentrations go up. But now you really see that insulin could in principle act virtually on all cells of the organism. And so approximately 20 years ago, we started asking the question, what could insulin do beyond those classical insulin target tissues to control physiology? And one of the regions which caught our particular interest was the central nervous system, because also the brain expresses very high levels of the insulin receptor as a principal insulin signaling component. So the brain is as insulin sensitive as the liver, the muscle, or the fat. And the question is now, what could insulin do in the brain? And this brings us back to an old hypothesis which was put forth by Kennedy in the 50s. And he postulated that there should be signals which communicate to the brain the energy availability of the organism. In other words, the brain should be instructed how much energy is around in the periphery of the organism. And he postulated that there should be hormones which are released in response to energy storage, and the receptors should be in the brain. And if there's enough energy, the hormones go up, communicate to the brain, and the brain makes you stop eating and to burn more energy to really maintain stable body weight. And this was a beautiful hypothesis, but it took another 40 years until Jeff Friedman's lab at Rockefeller University identified the prototypic regulator in this pathway as the hormone leptin. Leptin is released from adipose tissue. The leptin receptor is in the hypothalamus, the old region of the brain which controls food intake and energy expenditure. If you eat, leptin levels go up, released from fat. It acts on the brain, suppresses feeding, and increases energy expenditure. And so we hypothesized, could insulin, which is released from the pancreas in response to rising glucose concentrations, also act as an energy sensor, being signaled back to the brain to adapt these processes. And so basically what we generated more than 20 years ago were mice which had normal insulin action in all tissues of the organism, but the central nervous system. So they had normal action of insulin in liver, in muscle, in fat, everywhere else, but the brain. And now if we remove the insulin receptor only from the brain, the animals start slightly gaining weight, and they have a disturbed glucose metabolism. So this was really not consistent with the assumption that insulin only acts in liver, muscle, and fat to control glucose homeostasis. And so the questions that did arise from those early experiments was really how and where does insulin act in the brain to contribute to the control of systemic metabolism. And I would like to briefly introduce to you a very basic neuronal system which has been identified to be critical for the regulation of body weight and metabolism over the last 20 years by many groups, both in the States as well as in the rest of the world. So this is a scheme, a very small region in the hypothalamus, in this old region of the brain, which is critical for the control of feeding, which is called the arcuate nucleus of the hypothalamus. And there we have neurons which are characterized nerve cells which express a specific peptide, which is called pro-opiomelanocortin, or POMC. And those cells are activated by insulin or leptin when energy stores are full, and they release a neuropeptide which then acts on downstream neurons to suppress feeding. So if there's a lot of energy, insulin leptin levels go up, those cells get activated, and they make you stop eating. This is why they're called anorexigenic food intake suppressing neurons. There are neighboring neurons which do the exact opposite. They look alike under the microscope, but they release a different neuropeptide, which is called AGRP, and this strongly promotes feeding. These cells are inhibited by insulin and leptin. So it's really this yin and yang between those two antagonistic neuronal populations in control of feeding. And since those neurons are highly specialized to sense the energy state of the organism, we asked whether they maybe could also mediate the glucose metabolism regulatory functions of insulin through the central nervous system. And so this is really an earlier study by Christina Könner, a talented PhD student at the time. And so she removed the insulin receptor not from the entire central nervous system, but only from this either food intake suppressing POMC or food intake stimulating AGRP neurons. And what I really would like to point out, this population of cells in total encompasses only 3,000 to 5,000 cells. So it's only 3,000 to 5,000 cells in this very ancient brain region, which controls food intake in either direction. And when Chrissy knocked out the insulin receptor from either of those cell types, she found that the AGRP neurons, the ones which are activated in fasting, which are inhibited by insulin, mediate about half of insulin's ability to regulate hepatic glucose production in the liver. So this really brought us to a general scheme that insulin not only acts directly on the liver cell to suppress glucose production, but at the same time requires insulin action in those highly specialized energy state sensing 3000 neurons in the brain, and that both signals have to come together to fully regulate peripheral glucose metabolism. And so this really extends the picture that those neurons, which constantly monitor how much energy is available in the organism, that they not only control how much energy you will take up from outside of the organism, but also how you distribute your energy across different organs. In other words, how much glucose is produced in the liver, how much fat is released from fat, and they also control multiple other functions such as immune responses, bone mineral density. And what really fascinates us about those cells is that they seem to be the integrator which sense the energy state and then adapt the entire physiology of the organism to the actual energy state. And the big question, and this is very recent data, and I think it's also very much in line with what Andy has been talking about, the big question is how do those neurons, which in the scheme are located here in this arcuate nucleus, how do they actually get the insulin signal? Because there is not a very tight blood-brain barrier, but still those neurons are somewhat sealed from the periphery where this energy sensing takes place, or where the energy communication takes place. And so the big question has been in the field for a long time, how is insulin access controlled to those neurons? So how does the insulin get from the vessels to the neurons to control food intake, glucose metabolism, and so on? And what is highlighted here, this is the third ventricle. So this is a part of the brain where the cerebrospinal fluid circulates, and there are glial cells, specialized radial glial cells, which line the ventricle, and they send extensions next to the neurons, and they also send extensions to the blood vessels. And these cells are called so-called tenocytes. So these are specialized glial cells which communicate on one hand with the peripheral circulation, but also with the neurons. And so we asked the question, and that's a project by Marta, again, very talented PhD student in the group, who asked, could it be that insulin acts on those specialized glial cells to somehow control its action in the central nervous system? And so what Marta studied was exactly the role of insulin signaling itself in those specific glial cells, which on one hand contact the blood vessels coming from the periphery, and on the other hand contact the neurons that we had identified as critical regulators of feeding and metabolism. And what Marta could show is that insulin stimulates signaling in those tenocytes. If you remove the insulin receptor only from those tenocytes, insulin fails to elicit the signal. And very interestingly, if we have animals which are very obese and insulin resistant because we have fed them a high-fat diet, insulin loses its ability to activate signaling in those specialized glial cells. So this gives you two messages. Insulin acts on those cells, and under conditions of disease in obesity, insulin action in these cells is compromised. So the question is now, do we need insulin signaling in these cells for insulin uptake? And the answer is yes. If you inject a fluorescently labeled insulin into the tail vein of a mouse, it will be taken up by those specialized cells as they contact also the vessels, and they will pass it on to the neighboring neurons, which are in control of feeding and metabolism. If we remove the insulin receptor from these cells, they fail to take up insulin and to pass it on to the neurons. So really identifying them as critical gatekeepers for insulin uptake to those metabolism regulatory neurons. And what happens as a consequence of removing the insulin receptor from the tenocytes, these animals start gaining more weight, and they have impaired systemic glucose homeostasis. So really closing the gap between insulin released in the circulation and then being that it requires those specialized glial cells to pass it on to the neighboring neurons to exert insulin's regulatory function in metabolism. And I would just like to close with a small different vignette of relatively recent data, again, which were inspired through the collaboration with Andy's lab. What is generally accepted, and what I've been telling you about all the time now, is that these critical metabolism regulatory neurons are controlled by hormonal feedback, insulin, leptin. So in other words, hormones which are released from the periphery, which communicate the energy state of the organism. There is a clear prediction, if this would be the only mechanism how these neurons are regulated, you would predict if you eat, first the food has to be taken up in the organism, then it has to be stored in fat, then leptin levels go up, and then, for example, this POMC neuron starts firing to suppress feeding, and the AGIP neuron is shut off. But there was really pioneering work from many different labs, from both in UCSF and Brett Lowell's lab in Boston. And these groups could demonstrate that those neurons already start firing the moment the animal sees the food, sees and smells the food. In other words, the neurons behave already as if you have eaten just by seeing and smelling the food. And so we asked the question, what could be the role for this sensory food perception dependent transient regulation of feeding regulatory neurons? And this was work pioneered by Klaus Brandt, a postdoc in the group. And just to summarize the study, what Klaus could demonstrate is that the sensory perception of food was enough to transiently activate those food intake suppressing POMC neurons to regulate the liver and to exactly turn on that same pathway that Andy has been talking about, this XBP pathway, to prime the liver to increase protein folding capacity, to prepare the liver for what likely will be happening in the postprandial state. And so really to summarize what I've been talking about, those POMC and AGIP, so-called melanocortin neurons, coordinate the adaptation of integrative physiology to the energy state of the organism. AGIP neurons mediate insulin's ability to regulate hepatic glucose production, in addition to directly acting on the liver. In addition to homeostatic hormonal feedback regulation, they integrate sensory information to prime anticipatory physiological responses. Insulin receptor in those specialized glia cells, tenocytes, is critical for insulin access to the brain. Obesity causes insulin resistance in the tenocytes and thereby contributes to the dysregulation of systemic glucose homeostasis. And more generally speaking, in the context of aging, we think that those energy state sensing neurons coordinately regulate multiple processes that determine aging and the onset of aging-associated diseases. And with that, I would like to close to really acknowledge an amazing group of PhD students, scientists, and technicians. I have the privilege to work with many outstanding collaborators, really the strong collaboration with Andy. I had the pleasure to spend 2 sabbaticals in his lab, and a lot of the projects really have been developed having been inspired by the conceptual framework that I was exposed to, and of course our funding sources. And thank you very much for your attention.
Wow, thank you so much, both of you, Jens and Andy. That was absolutely amazing. And I think we can say for sure that it's great to see the international collaboration. You both showed groups and groups and groups of people that are working on it. You mentioned before that there are a lot of groups across the globe actually that are researching the topic. But I think it's also worth mentioning one very unique thing about the German system. And Dana is the representative of the DFD, the German Research Association, and this is they allow with their grants that they give out for actual time to do the research. It's well-funded research, and it's not rushing anything, and it's not looking for the next fast application, but you actually have the time for discovery and also the time to do basic research. I think this is a former student of mine mentioned that before. This is very unique in our system. So this is a fantastic example. Thank you again. I'm very psyched. I have to say I understood a lot, contrary to what I thought before, because I'm not from the field. But I do have I have two burning questions before. By the way, the floor is open to all of you if you want to put your questions in the chat. And we go over time a little bit, if you don't mind. If you can stay with us, Andy and Jens, that would be great. My questions would be, so you have all these really groundbreaking results already and findings and very robust findings that you've done with your experiments in worms and mice also. But of course, I think we all have in mind, being humans and being somewhat egotistical probably, what is the next step then? First of all, what is the next step for you? Where are your groups going to? Where are you going? And then also, how can that be scaled? And will it take long? Maybe the question's maybe too big, but maybe you have an indication.
Maybe I can go, just start out. And so basically what we're trying to do at the Institute, and I think this is also, I mean, again, what's going on in CICCAT, that it's really spanning, multiple organisms, what we're trying to study. And so as soon as we, identify a mechanism in mice, or even coming, earlier from worms, what Andy has really put out as a true basis of many of those experiments, we really also have the setup for truly translational studies. And that, of course, is difficult, right? If you want to study, let's say, neuronal control in humans, it's inherently difficult to assess this, right? And so there are imaging technologies such as functional MRI imaging or PET imaging, with which we can at least try to validate whether the same principles hold true also in humans. And this is exactly what we're trying to do, is to, take the hypotheses that we're generating in mice, the predictions, and try to see as much as, even with the lower resolution of technologies that we have available and which are, pretty demanding. For example, the tenocyte business. So now we show that in mice, if you get obese, you have less insulin access, and that regulates the excitability of those neurons, which are critical for metabolic homeostasis. And the next step is now to try to, generate radioactively labeled insulin, try to validate that we can image the transfer of that to the brain in mice, with the ultimate aim to use that same tracer to then study lean and obese human subjects, whether the same principle holds true. And that's kind of the philosophy that we're following. But as you can imagine, I mean, it's a long way. You cannot do everything that you can do at the high resolution in such a model system. But at least you can, try to prove or disprove the fundamental principles that you have just covered also in the human situation.
Andy, what's in it for you? What's the next step for you there? Thank you, by the way, Jens. That of course makes a lot of sense.
Yeah, no, great presentation, Jens. I think, that this whole, I think I'm totally fascinated that aging and metabolism is not entirely cell autonomous. That would have been a disaster if we were going after translational applications, that you would have to hit every cell in the body. So it's a blessing and a curse, the discoveries that Jens and I are finding in the rest of the field, that this neuronal control, we only have to hit a few neurons or a few glial cells and get these dramatic peripheral responses, is really fascinating. It's good therapeutically, but as Jens said, hitting those, getting into the nervous system and hitting this is going to be difficult, not undoable, but difficult. The thing that, is insulin the whole story? From Jens's work of these tenocytes passing it off, we need to figure that out as well, even using the worm C. elegans. It has an intact insulin signaling system. Or are there other molecules to be found? And if we find those other molecules, are they themselves going to be therapeutics? Can we actually get around this business of stressing out these neurons and these glial cells and just giving these hormones, so to speak? And I want to say that with caution, because, the presentations that you've heard from me and Jens, the 20 minutes from each one of us, they're all the positive results, all the things that are working. We don't know all the negative downsides that may happen. We haven't looked at everything. We don't know turning on these response pathways, does it predispose you to cancer? You know, is there actually some aspects of neurodegeneration that may be associated? So I don't want to jump so quickly that we are going to figure out a therapeutic for aging or even, you know, most forms of metabolism yet. I think we still have a lot of work to do. So I want to put caution on it. Sorry to put the fire down a little bit.
Yeah, but it's the same.
No,
I mean, just as a small, small addition to that, what Andy is saying, I think, like, because what we're studying is a highly specific neuronal network, right? I mean, what we're targeting is 3,000 cells out of millions of neurons. And now, if you would flood the brain with high levels of insulin, it would act on other neurons. And we know that even in other cells, overactivation of insulin signaling can do the exact opposite what we want to achieve, right? So it's really critical to find ways, and insulin clearly is not the sole response to that. It's just, but how can we modulate specific neurons specifically? And that is a vast challenge, which I think we're really far away from, getting to translational.
Thank you so much. Of course, always with caution, and clearly it takes more time, but I think what you've shown today already is a huge step forward. So I think there are leaps that are being taken. So I wouldn't expect to see anything on the market next year, but I'm sure that a lot of people are paying close attention to your research to figure out, can we produce something and find solutions. But we have a few questions in the chat, if I just may pose them to you. One is, are there any pathways which you're studying in regard to the blood-brain barrier? John is asking.
Yeah, I mean, this is exactly what this tenocyte comes down to. And what I should point out is that our initial hypothesis was that we need the insulin receptor on the tenocyte to really pass on physically almost insulin from one side of the brain to the other, let's say, right? But what we have found is if we modulate the insulin signaling in those gatekeeping tenocyte cells, it not only affects insulin uptake, but also the uptake of other hormones. So I think it's not only the insulin being passed on through its own receptor, but that insulin does something very fundamental to that blood-brain barrier, which not only controls insulin access, but also that of other hormones. And so I think if you're again talking about your translational perspectives, if we now understand that those cells become insulin resistant, maybe if we can resensitize these cells for insulin, it could reinstate the normal transport, not only of insulin itself, but also other hormones to exert their intact regulatory function. So I think those are kind of the philosophies. If we understand why in disease does that regulation not work anymore, maybe we can find sensitizers which reinstate the normal insulin signaling as one example to reinstate the global normal functionality of that cell type to help improve metabolism and neuronal communication in general.
Would you like to add anything, Andy?
I think it's so, Jens and I have great collaborations, but it was amazing that both of our labs independently came across glial cells, independent of each other. And it was like one day we were talking and I told him about Ashley's work and he said, "Oh my God, we have these tenocytes that are doing a similar event." And we crossed paths again. So I think it's just, it is, I think, a new area of neuroscience. And I also think it's definitely a new area of aging research, that glial cells can be the major drivers of many of these processes. And maybe they are the gatekeepers of which hormones get presented to the nervous system. I think it's just a totally fundamentally interesting idea, just for basic science, not just for aging and metabolism. I think it's just a fantastic idea.
Great. And I think we have time for maybe one or two more questions. There's one from Tatiana. And Tatiana is saying that I have a question about the fasting and hormone levels. What has happened to leptin levels and insulin levels during fasting? How does the PMSC and the HRP neuron react? And do they become insensitive to the repetitive stimuli? Do they stop firing or what?
Basically, I mean, so the normal dynamic regulation is if you're fasting, your insulin and leptin levels drop. And then those HRP neurons, which are normally inhibited by those hormones, become disinhibited, in other words, activated. So if you're fasting, insulin, leptin levels drop, the AGRP neuron. So basically, your body communicates to the brain, there's not enough fat, there's not enough glucose, and then the AGRP neurons start firing, and they drive you towards eating and to produce glucose from the liver.
I hope that answered the question. And then I'm taking the last one here. My question is about NMN. We're about to get a storm in the US for the supplements because author Tony Robbins has it in his last book about anti-aging. What are your thoughts on that?
You talk about nicotinamide mononucleoside, NAD supplements. I mean, the data looks, it's pretty good in some of the lower organisms. I worry about, you know, when you flood your system with as large doses of NMN, you know, what tissues are they getting to? And, you know, I don't know what other side effects maybe, you know, we should be worried about. You know, it's early days on NMN.
Any thoughts, Jens, or none at all? Okay, I think that's fine, and I think this is kind of a funny way to wrap up. But we're a little bit over time already. So thank you so much for this super interesting insights and for giving us a glimpse into your highly important research. It was wonderful listening to you. It's very, very engaging. And also thank you to you for joining us and for staying a little bit longer. Thank you to the University of Cologne and Eva for putting up the Transatlantic Tandem Talks. Keep doing that. We will be your partner all the way. And also a big thank you to the DFG and Dana for making such amazing research possible. Thank you. Have a wonderful afternoon and evening and maybe start of the day if you're joining us from the West Coast. And we hope to see you soon. Thank you.